In this 7th part of our video seminar series on mitochondrial heat production, we'll look at the potential implications of hot mitochondria for health and disease. Last time, we looked at the implications for evolution, including the origin of eukaryotes and of homeotherms, and the question of whether bacteria might also be hot. I'm going to start this segment by revisiting something we mentioned briefly last time: the possible connection between mitochondrial genetics and adaptation to climate, leading us on into the area of nutrition and obesity, and then on to mitochondrial pathologies more generally, as well as of the heart specifically. The human mitochondrial DNA encodes just 13 crucial polypeptides of the OXPHOS complexes. But soon after it was first sequenced, when it was established that it evolves 10 times faster over evolutionary time than nuclear DNA in humans, at least at selectively neutral sites, it was realized that the mitochondrial DNA of different ancestrally linked populations had distinct mitochondrial DNA haplotypes. These can be used to trace matrilineal ancestry in relatively recent time. The mitochondrial DNA haplotypes are distributed globally in specific geographical regions, with little population mixing until recently, and can be classified into geographically and ancestrally related clusters or haplogroups. Haplogroup L, for example, is found almost exclusively in Africa and persons of African matrilineal ancestry, wherever they live today. The Inuit, originating in East Asia, took their East Asian haplogroups with them when they migrated to the Arctic region of North America. However, at a deeper level, the specific mitochondrial haplotypes in the peoples of the Arctic have been proposed, in some cases with experimental support, to shift the mitochondrial energetic balance between ATP and heat production so as to favor survival in a frigid climate. These issues are discussed in much greater detail in the papers referred to on the slide. Note also that there is an additional dimension to this apparent trade-off, which is that burning of surplus substrate under conditions where both ATP and heat production are sufficient or even limiting results in enhanced biosynthesis, notably of lipids. Mitochondrial haplotypes that are adaptive under one set of conditions, for example cold climate and limited nutritional resources, can be deleterious in another, leading, for example, to obesity or to disorders in which overproduction of reactive oxygen species is a contributory factor, and this includes both cancer and neurodegeneration. The range of symptoms and severity of mitochondrial diseases, even just of those caused by identifiable mutations in components of the OXPHOS machinery, is vast and perplexing. The explanations advanced for the various clinical manifestations include at least those listed here: insufficiency of ATP supply, redox imbalance, overproduction of ROS, disturbances in calcium homeostasis and that of other ions, as well as diverse possible effects on metabolism. Metabolism being so complex, interlinked, but also having tissue-specific aspects, there are many ways in which this impact can be felt, such as via the TCA cycle or 1-carbon metabolism, as illustrated. But derangement of mitochondrial heat production, or its regulation, has thus far been only rarely considered as a factor. This despite the fact that the very first mitochondrial disease to have been characterized in the 1950s and early 1960s by Rolf Luft and colleagues was actually a case of malignant hypothermia associated with muscle weakness in which mitochondrial respiration was largely uncoupled from ATP synthesis. Other isolated such cases have since been reported, but in principle, a disturbance in the mitochondrial heat ATP balance, or simply a decreased rate of respiration, could also lead to an opposite phenotype of hypothermia. And there is evidence of that too in specific cases, although it has not been systematically explored. I obviously think it should be looked at closely, as well as in animal models, now that we have suitable tools. In fact, I think some relevant data on this may be published quite soon. An important corollary to this discussion is that in many cases, the symptoms of mitochondrial disease first manifest or are significantly exacerbated by episodes of viral infection accompanied by fever. This has been seen in the case of many point mutations in mitochondrial DNA that manifest as muscle disease, characterized by the classic feature of ragged red fibers, or as sensorineural deafness. A reasonable working hypothesis is that the mutations predispose to excessive mitochondrial heat production, but this is tolerable under normal physiological conditions. However, the additional thermal stress occurring during fever is sufficient to push the subject's mitochondria and/or other cellular systems over the edge into overt dysfunction. I'll now turn to the specific example of the potential role of mitochondrial heat production in heart disease. Although some of my best friends and colleagues are cardiologists, I am not. So the treatment of the topic here will be simplistic, not to say simple-minded. However, I do believe that the basic concepts are valid. Sometimes simple ideas are the best. Essentially, the heart is a machine that pumps blood to and from the lungs and to the tissues in general. Like all biological processes, this pumping action requires energy, initially in the form of ATP, derived from mitochondrial substrate oxidation in the heart muscle cells. As we considered repeatedly in parts 1 through 4, the biochemical reactions of OXPHOS are far from being 100% efficient thermodynamically, up to half of the input energy being routinely converted to heat, not ATP. Moreover, in energizing biological processes such as muscle contraction that enables the heart to pump, the use of ATP to do work is also not 100% efficient, and is therefore once again thermogenic. The heart is one of the body's major users of energy, so where does all this excess heat go? The answer is clear. It's carried away in the bloodstream, helping to maintain the overall body temperature, along with supplying the nutrients and oxygen that the heart also supplies to the tissues. This should be seen not only as a way of saving the heart from overheating, but also of supplying heat to the rest of the body. All kinds of things can go wrong with the heart pump, or to damage it, including, of course, primary mitochondrial dysfunction with a drop in thermodynamic efficiency of OXPHOS, but many other insults, including physical limitations on blood flow into or out of the heart itself. Under such circumstances, the pump has to work harder to deliver both oxygen and heat. But as it pumps harder and harder, more and more heat is produced, leading to a vicious cycle. Eventually, the pump does overheat and a catastrophic failure can ensue. Obviously, this does not explain all forms of heart disease, but I believe it is a plausible common scenario. Resolving the problem is, of course, not at all straightforward, but somehow installing an accessory cooling system might break the cycle. Food for thought. I'd like to take a brief detour at this point to discuss some of these issues in a little more detail with my colleague Mugen Terzioglu, who has been working on some relevant aspects of mitochondrial heat production in cells and in flies. So, Mugen, if mitochondrial dysfunction so frequently leads to overheating or overcooling, why has that been ignored, not looked at even by clinicians until now on a wide scale?
So we know that mitochondrial diseases are really rare, and we have trained, practiced clinicians to diagnose them. And usually clinicians are always taking consideration to look for mitochondrial main function, which is ATP production. And ATP, if there is an ATP deficiency, if there is a complex functional losses, these are the major things that all clinicians are biased to look at. So there is actually a lot of literature about heat production, mitochondria, and clinical records. So this is major problem for heat overlooked as a clinical determination.
So, in my presentation here, I've speculated that mitochondrial heat production is a key factor in heart disease and also in neurodegenerative disease. So, are there other disease categories that we should be looking at or should be considering mitochondrial heat production as a crucial factor?
I think yes, because most of the metabolic disorders, they cause metabolic dysfunction, and it should be also considered like diabetes, immune deficiencies, leukemias, and even cancer should be reconsidered in terms of mitochondrial heat production and deficiencies.
It's now time to preview the next video in the series, when I'll look at some of the objections and caveats that have been raised to question the hot mitochondria hypothesis. As always, the jury is still out. Have we made erroneous assumptions? And are our measurements somehow invalid or theoretically impossible? Or have others misinterpreted the laws of physics? We will look at various leads coming from nanoscale physics, the study of respiratory supercomplexes, and the actual composition of the OXPHOS enzyme complexes themselves that might help us address and resolve these issues. Finally, I must thank once again my colleagues, collaborators and funders, notably Martin Sibor, who has opened my eyes to the complexity as well as the simplicity of the heart, and Pierre, Paul and Mugen, who have contributed greatly to the thinking on mitochondrial pathology behind this video in the series. Thanks also to those whose images I have borrowed.